Nice to meet you everyone. Today you are here with me, Prasita Chanchai, and my partner, Thaladi Hawasotwong. Today we are going to present our project in topic of analysis of total phenolic content and total flavonoid content in product from raw banana powders. First of all, we need to know about the word free radical, which are unstable atom or molecule that can damage the cell. So that we have antioxidant, which are substance that can use to prevent or use to slow down oxidation reaction. For example, substance in phenolic group, flavonoid and carotenoid. And what are phenolic compounds? Phenolic compounds are a substance that have one or more hydroxyl group on aromatic rings, and we can divide it into three major groups. The first one is simple phenol. The second one is phenylpropanoid, and the last one is flavonoid. And we can find this kind of substance in very kind of fruit and vegetable. And one we should to study is banana because it's a well-known fruit that have many bioactive compounds and beneficial substance, which including phenolic and flavonoid too. And we can see that some research say green banana show higher total phenolic content and total flavonoid content value than those dairy food. And about the research about raw banana powder, we can see that it's usually used to help killing acid reflux and also some used to lose weight. But the original but we never seen a project that studied total phenolic content and total flavonoid content in raw banana powder before. And foreseen that this study may be a helpful information in the use of raw banana powder for more usage and development in a vegetable area.
So the hypothesis of this project is the selected banana products may contain sufficient amounts of total phenolic content and total flavonoid content, which may provide potential antioxidant activities. For the objectives, there are two objectives in this project. But the first one is to study total phenolic content and total flavonoid content in raw banana powder. While the second one is to study whether raw banana powder can be an antioxidant health supplement in addition to being an acid reflux medication. So these are our equipments and materials. For the methodology, we have divided the methodology into 4 parts. But the part 1 is sample substance preparation. First, weighing raw banana powder 1 grams, adding 5 milliliters of methanol, and then mixing it in the vortex mixture. Extracting it using ultrasonic bath for 5 minutes, and then spinning it in the centrifuge for 1 minute. Repeat only the supinate tank, which is the liquid part. So we get the sample substance 200 milligrams per milliliters, and then dilute it for 5 concentration. For the first one, we dilute it by 2-4 and then we dilute it by 10-4 from 200 milligrams per milliliters until it is 0.1 milligrams per milliliters. And the second one is standard substance preparation. We use gallic acid to compare with the total phenolic content, and we use quercetin to compare with the total flavonoid content. We use gallic acid 10 milligrams, adding distilled water for 10 milliliters, and the quercetin 10 milligrams, adding methanol for 10 milliliters. So we get the stock gallic acid and stock quercetin in the concentration of 1 milligrams per milliliters, and then we dilute it by 2-4. Dilute it for 11 concentrations from 500 micrograms per milliliters until it is 0.24 micrograms per milliliters. Third one is analyzing the total phenolic content. We use sample or standard substance, which is the gallic acid, 20 microliters in the 96 well plates, adding folate CaK2 reagent 100 microliters in each well. Then adding the 0% sodium carbonate 80 microliters in each well. Incubate for 30 minutes. Measure the absorbance by the wavelength 750 nanometers in the micropet liters. After that, we analyzing and plotting the standard curve. For the last method is analyzing total flavonoid content. We use sample or standard substance, which is the quercetin 100 microliters in the 96 well plates, adding this 5% sodium nitrate 20 microliters and left it under the aluminum foil for 6 minutes. And then we add the 10% aluminum chloride for 35 microliters and left it under the aluminum foil 6 minutes again. After that, we measure the absorbance by the wavelength of 430 nanometers in the microplate leader, and then analyzing and plotting the standard curve. So these are the results. For the figure 1 is the calibration curve of standard gallic acid, which is used to compare with the total phenolic content, and for the figure 2 is the calibration curve of standard quercetin, which is used to compare with the total phenol content. And these are the results. For the first one is the total phenolic content from 10 samples. As you can see that all of the raw banana powder samples have the total phenolic content in it. And number 10, which is the in-house raw banana powder, has the most total phenolic content. And these are the total phenolic content from 10 samples. As you can see again, all of the banana powder samples have total phenolic content in it. And number 10, which is the in-house, have the most total phenolic content from all of the raw banana powder. For the conclusion is the analysis of the total phenolic and total phenol content in this experiment suggests that banana powder may have good antioxidant efficacy, which can be developed in antioxidant health supplement in the future. And that is for today. Thank you for watching and listening.